Okay, so on behalf of my fellow organisers, Yelena, Osama and Layla, who you might see up at the top there, I'd like to thank everyone for their attendance today and welcome them to this new seminar series. We'd also like to thank the Society of General Physiologists and Jean and Greg in particular for their support in setting up this series. As everyone here probably already knows, then this series is a platform for junior faculty and postdocs to present their work in the broad field of electrophysiology and ion channels. And so these seminars will take place on the first Tuesday of every month at this time. And we're very excited to be putting together the schedule, which can be seen in full as it develops at the website, which is in the chat, www.sgp-epic-seminars.com. We're also very happy to receive further nominations for speakers, which can come from mentors or colleagues or self-nominations as we continue to develop the schedule. And one feature of this series is that each speaker will be invited to host the following month's speaker for a virtual visit, where one-on-one meetings with local interested researchers will be organised. And so we hope this will promote new connections amongst these young investigators. And so we also hope to span a wide range of topics from biophysics and structural biology through pharmacology, physiology and disease, and to showcase some of the most exciting recent work in the field. And so with that, I think it's very fitting that our first speaker in this series is Willow Coyote Maestas. Willow is a QBI and HHMI Hannah Gray Fellow and begins as an assistant professor within the Department of Bioengineering and Therapeutic Sciences at the University of California, San Francisco, I think officially in July. So Willow completed his PhD at the University of Minnesota under the supervision of Daniel Schmidt, where he was recognized for his thesis work by numerous awards. And so Willow is developing multiple cutting-edge approaches, including high-throughput deep mutational scanning and insertion methods to investigate membrane protein structure and function. And just to prove we don't discriminate too much here, I think you may even talk about some transporters and GPCRs, right? So we're very inclusive. So with that, I'm delighted to hand over to Willow, and just a quick note, please feel free to post any questions in the Q&A box for discussion at the end of the seminar. And alternatively, if you would prefer to give your question personally, if you raise your hand with the raise hand function, we will try and introduce you in the Q&A seminar at the end. So with that, I'll hand over to Willow. Welcome, and thank you very much for joining.
Thanks so much, Connor, and everyone for giving me this opportunity and the kind introduction. As Connor said, I'm just starting. I've been at UCS for a bit, but I'll be starting as an assistant professor in a couple months. This will be my first talk since I made that decision and all of that. So I'm very excited. And I promise I haven't completely forsaken channels, and I'm going to continue working on them. And I'll talk a little bit about channels, but I'm also very excited about transporters and GPCRs too. So I love all membrane proteins. And so what I'll talk about a bit is the mechanistic genetic screening approaches that we've really been using to push forward and uncover what I like to call the secret life of membrane proteins. And that's a fancy way of just saying, you know, beautiful, cool membrane protein biology. And so I've really fallen in love with membrane proteins, as many of you probably have too, since the beginning of my PhD with Daniel Schmidt, where I really appreciated that they serve as the interface between the chemical and physical environment around a cell. And this allows us to, of course, this molecular role allows us to have complex sensory physiology so we can perceive the world, so you can see my slides. And then, you know, complex electrophysiology, as many of you will love, that hopefully that allows me to conceive of my science and perhaps you to hopefully understand a little bit of it. And then, of course, membrane proteins serve as the interface, which means they're targeted by a huge number of pharmacologies. And mutations within membrane proteins have enabled this immense diversification so we can have complex electrophysiology and physiology. But unfortunately, these mutations, of course, cause disease, the diseases that ail us, or disrupt the drugs that we use to try and treat these diseases. And there are many different mechanisms by which a given mutation can give rise to its impact, from the biophysics of folding to the cell biology of trafficking to even the structural biology of function. And this has meant that we have been so hampered in the membrane protein field because Conventional approaches in membrane protein biology, of course, as many of you will appreciate, are tremendously challenging and slow. Beautiful, say, electrophysiology, as many of you do, that I'm in awe of, and I can't do myself. And I'm like, wow, you guys are so good at what you do. I'm not a good scientist at all. Or structural biology, all these things are so slow. And that means we can't know how every single mutation that exists within all of us could give rise to disease or disrupt pharmacology or enable us to evolve new functions. On the other hand, there's a number of high throughput methods, some of which I've developed and used, and typically they're really not mechanistic enough to really learn how variation alters membrane proteins. And so that means we're really limited in understanding how membrane proteins fold, how they traffic, and how they function in physiology and how they cause disease. And so my solution to this, and what I've been working on and what I'm excited to continue pushing forward, is really what I call mechanistic genetic screening of membrane proteins. And what I mean by that is if we think of a genetic screen, typically this could be, say, a CRISPR screen, which some of you may have done, where we knock out all genes in a cell, or a missense mutational scan, which is a lot of what I've done, where we make every single point mutation. But typically these sort of assays are hooked up to some cell fitness phenotype or cell growth. And while these assays have been tremendously important in identifying genes that are important in all sorts of physiology, particularly in the membrane protein field, of course, for a gene to play its role and for a membrane protein to play its role in cell physiology, it needs to be translated, it needs to fold, and yada, 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 until you get to physiology. And if we just measure the top here, if we just measure cell growth, we're leaving so many phenotypes on the floor. And we're losing so much mechanistic information. So my dream, or where I want to go, is to be able to make all sorts of perturbations, but measure all the way they can alter a protein. And then we can really understand holistically how a mutation or a CRISPR knockdown breaks proteins to understand disease, but also so we can understand basic biology. And so I'm going to tell you how we're getting there and where we're at currently. First, I'm going to tell you about the foundational technologies that we're using to increase the sort of perturbations we can make, to explore more on the x-axis in that plot. And then I'll talk about how we applied this in a model potassium channel now a while ago, but that allowed us to feel, or allowed me to feel, more confident in going into new spaces because we were able to validate and extend structure function models and then push forward clinical genetics. And then finally, we've applied this in less model systems such as drug transporters to really start understanding how they work and how they function, and maybe even pushing forward pharmacogenomics eventually. But first, we'll go to the foundational technologies. Perturbations sit on a continuum here when we think of perturbations in life, but also in our genetic screens. And so the distinct ends of this, as I said before, are really this missense and CRISPR screens where we make point mutations or we knock down genes. In my PhD, I did a tremendous work on protein engineering where we were inserting whole domains into every single position of a protein. We were using that to engineer light switching for optogenetics, but also so we could start studying how regulation evolves in a protein. I'm not working on that as much anymore, so I won't talk about that too much. Also, it's all published, so you can read the papers. But when I started to switch over to doing missense mutational scans, because I was interested in what is more relevant perturbations in the context of evolution and disease, I found that there's this huge gap between changing the physicochemistry and asking the question, how does this residue contribute to a fitness phenotype, versus inserting an entire 10 kilodalton domain to every single position, which told me something about the backbone flexibility of a protein. So when I started at UCSF a couple of years ago in this position, I was really wondering, what are we missing here? Doesn't matter, because there's just such a huge gap here. As it turns out, insertions and deletions, or short sequences that are being inserted or removed from a protein, are tremendously common in disease, are really important changes in evolution, and they're really understudied. And of course, in the membrane protein field, folks have been using deletions for a really long time, such as Lily Jan, first with this ER trafficking variant that was used to identify how these channels make it out of the ER. And then Paul Welling identified this Golgi trafficking motif, which is used to get out of the Golgi, the surface, and it's probably involved in folding too. And we can measure at scale how proteins make it to the surface if we had libraries of deletion, so we could search, say, for delete sequences that are involved in trafficking at scale. But unfortunately, we really don't have good methods for making indel libraries. So these are the 3 papers that were published before we did this work in this space. A beautiful paper from George Church's lab on AAV engineering, an antibiotic resistance paper on by Mark Ostermeyer's group at Johns Hopkins, and the Benedetta Bolognese's lab, a beautiful protein on or a paper on a amyloid generating protein. But this is it. Across like all proteins that I know of, these are the systematic studies that have been done. And as I said, this is because we don't have ways to make libraries that are generalizable that include indels. So when I started at UCSF, this was the first problem I wanted to solve so that we could start making these libraries for our experiments. And I worked really closely with a tremendous postdoc in Jamie Fraser's lab who joined, and I kind of distracted him from his main project, but he got a paper out of it, so yay. And then a fantastic grad student, or rather a good friend of mine who was a grad student in Daniel Schmidt's lab together. And then after he left, in the evenings and weekends, he would continue working on this project because he's just so generous, David. And then a fantastic tech who joined my group, and a grad student who also joined, who worked all together to do this. And so the basic idea is we take a gene, we break it up into parts, and then within that part, we synthesize oligo pools that are complex that have all the mutations we want within that part. And then we can add on cut sites or restriction enzyme sites that remove themselves, called type 2S restriction sites. And we can use those in Golden Gate to replace this region of a gene in the plasmid with a mutated region. And then we just break up our gene into parts. So effectively for CRU 2.1, 400 amino acid or so gene, we break it up into 8 parts. And that's because there's a limit in the length of oligo pool you can order. And we call this deep indel missense programmable library engineering, or DIMPLE, because it makes us happy. And so I made this cute little cartoon because I thought it was fun. But what matters is, does it work? And so this is looking across a gene, CRU 2.1, at the amino acid positions on the x-axis. And then the y-axis is how many mutations are being made per position. We can see it's tremendously even for the vast majority of it. There's no lack of coverage. And actually, this kind of drop-off is more due to sequencing and not our libraries. And then we wanted to ask the question, we want to have all variants equivalently incorporated. And what we find is that all variants types are incorporated at similar frequencies. Insertions are slightly less depleted, probably because they're making things longer, and there's a fitness cost to that in bacteria, but it's not substantial. It's all within a fold or so. So all variants can be incorporated at all positions. And then we tested this on a number of other genes, and indeed we can find we can make these libraries in many cases. We've done this many other times. Folks around the world and the country are doing this as well. This approach works, including TRPV1, which I think this has been like my great white whale. I've been trying to work on it forever. I'm sorry I don't have data on it because it's a challenging gene, as some of you may know, but one day I hope to present data on this. But we were able to make a library at least, and it's quite big. But so in CRU 2.1, disease mutations are known, including deletions, to cause developmental and cardiac disorders. So what we did, just to show we could use this library for something and learn something new, is we applied this library to our pipeline, where we take our gene, we make a library, as I showed, and then we can make stable cell lines that are HEC293 cells, which every single cell gets a single copy. And then we sort based on surface expression using a fluorescent antibody. And then we can sequence out and then calculate fitness scores. So this is what that looks like. This is a highly rich data set. So this is across all of CRU 2.1, all 436 positions, it looks like, and on the x-axis. And then the y-axis are the variants that are being made. So at top, there's a bunch of missense variants, and then there's insertions and deletions that are separated. And we can see very clearly that there are strong patterns where there's red regions, or regions that aren't making it to the surface. And there's far more red regions in the deletions than insertions, than even the missense variants. And you can kind of see this diagonal pattern across, which implies there's different kind of regions that are structurally sensitive. And so, in other words, there's a perturbation hierarchy where, so now we're looking over here on the right, and here synonymous mutations are centered at 0 in this plot. So this is surface expression fitness, so they're all expressing like wild type. which makes sense. We're not changing the physical chemistry. Whereas missense variants change the physical chemistry, and you get some that increase. So this is looking across all missense variants. We see some that increase surface expression and many that decrease. And then insertions we see are more disruptive. There are far more that are disruptive and a handful that are more. And then deletions are more disruptive. So that makes sense. Insertions are pushing things apart and deletions are wholesale removing sequences. And over time, as we do more of these experiments, we can learn rule sets and learn how they break. But finally, we wanted to start exploring how pathogenic variants in the context of the screen, what they look like. And so this is now looking at all insertions and deletion scores, the mean of all of them. So across the gene, and we can find that there's a number of regions that have low expression. And of those, and there are 4 patient deletion variants. And of those, 3 of them have low surface expression. So if we just had this data set, we may think to ourselves, oh, wow, this Delta 308 or 306 version isn't a pathogenic variant. As it turns out, that's in a functionally critical region, the G loop of this channel, that I'll talk about in a bit. But by just measuring surface expression, we really miss out on that. So if only we could do that. So overall, we find that 3 out of 4 known variants, and there's probably many more in the population, don't contribute to surface expression. So overall, with Dimple, we have this really robust pipeline that allows us and others, everything's open source, we have the code, we have a protocol that we tried to make as detailed as possible, and we're continuing to refine it to make it easier, that other people can use to make similar libraries. We've used these to start exploring how the impact of indels impact different parts of secondary structure that I didn't tell you so much about, but over time we hope to learn how insertions impact membrane proteins to guide evolution. And finally, we can start inferring what is the impact of indels in the clinic to understand this important yet wildly or massively understudied class of mutation. So next, I'm going to talk about how we then use these sorts of libraries to start really learning about structure function and pushing forward clinical genetics even more. And so, as I said, in CUR 2.1, we know there are disease mutations that break trafficking and folding, but there are also disease mutations that affect function, of course. And so this is some mutation that's found in a patient that is surface expressed fine, but of course doesn't function. And so when I set out on this project in Daniel Schmidt's lab at the very, very end of my PhD, in the last 2 months of my PhD, in fact, I was living here in San Francisco as a visitor, and I flew back for a week to do this experiment because I just really wanted to see how this worked, because I just couldn't wait. And so what we did is we took a gene, CUR 2.1, and we made all the missense variants using an earlier iteration of our pipeline spine, and we made stable cell lines again. And we use an assay for surface expression using a fluorescent antibody, and also an assay that's messier, but for function, using a voltage sensor dye used in plate reader assays. And then using that, we could sequence out and get at the surface expression and the functional fitness of all the mutations in the library. So again, we can look here at the surface expression phenotype. And so it's very similar to what I showed you before, or synonymous mutations are centered at 0, and now missense mutations are greater than or less than. And this was before we started having indel libraries, if you're wondering where those are. This is a very similar heat map to what I showed before. If you're confused, if you're like, wow, the colors are swapped, you're right, they are. And the reason why is because the postdoc in my group really wanted to switch the colors on us because he liked red to be lower and blue to be higher. He's doing the work, so he gets to decide where things end up, even if it confuses people for my talks. But what we can see is that known motifs are something like the flag tag motif, which we've inserted to allow for antibody binding, is highly sensitive to mutation, or is blue, which makes sense, doesn't make it to the surface. Golgi export motifs that are well known, some of which I showed before, and I think some of these are involved in folding too, are very sensitive to mutation. And then an ER export motif is less sensitive, but there's even a little gain of function mutation in between. And if you're a very keen observer, you'll see that there's these two acidic residues on either side. So this is a classic diacidic motif. And in fact, if you make a third acidic mutation in the middle, it boosts surface expression even higher. So that's exciting because maybe triacidic is better. Interesting design thing we learned, just like kind of a side observation we'd want to test more further. We can take this data, we can map it onto the structure. It's a tetramer, of course, like many channels, but it's easier to look at it as a monomer. We can find that there's two strong blue clusters there. And to take a somewhat unbiased approach to trying to understand what those might be and why they might be driving surface expression, I just asked the question, where are there hydrophobic or aromatic amino acids that when you mutate them are highly disruptive? And then just map those onto the structure in surface filling mode. And there's two different colors because I just want to highlight two different regions of the protein. And so we see these two strong clusters of residues that are driving folding that are driving surface expression that are hydrophobic, so are very likely folding determinants. If we look at the top one, what is that? That's this classic pore loop or pore helix that many of you are going to be familiar with, of course, that Carol Deutsch's lab studied with really laborious, slow, hard in vitro studies of folding, which are very beautiful, and a couple of papers in alanine scanning mutations. So I'd like to point out to you biophysicists that we did this in a week for all the mutations versus having to do individual ones, but your guys' work is much more elegant than mine, but, but haha. But we were able to do this really, beautiful systematic study, I think, that allows us to identify these residues and perhaps even develop more deeper design principles if we did this in other backgrounds too. And then there's the other ones, of course, this hydrophobic core of this immunoglobulin-like domain that some folks had characterized in thermodynamics in Kerr 1.1, but we're able to systematically identify which residues, say this tryptophan, are more important versus, say, these valines, which are less important, but they're all apparently important. You see it wraps all the way down. And I think this is so beautiful because you have the top and the bottom are involved in folding, and the middle isn't at all. So what is that doing? So we did our functional fitness assay. So this is using a voltage sensitive dye that partitions into the membrane when the cell is more positively charged. And of course, Kerr 2.1 sets the resting membrane potential around -80 or so when it's expressed by itself. And so we're able to see a shift in this dye to be less fluorescent, and then we can gate on that. It's a very messy assay, relatively speaking, which frustrates me, but it's just how the cookie crumbled. But we can still see synonymous mutations are centered at 0, and missense mutations, some are greater than and others are less than. We can look at the heat map. I won't spend a ton of time on this because, again, it's very rich, but it broadly follows the same trends as before. And really the problem here, of course, is channel function is going to be the combination of how many channels are at the surface and how functional each is. And so if we want to understand which residues are contributing to function alone, we have to remove the number of channels. So we simply just took these two data sets and subtracted them and took the mean to identify which residues are involved in function. And that gets us the somewhat abstract but meaningful and interesting score here. So we're just taking this data set and subtracting it from this one. So just to ground ourselves, if surface expression is dominating because the range of the data sets are different, this ends up being a slightly positive score when this is z-scored. And so this is all these residues in blue are contributing to folding or surface expression. And then all these residues in pink or magenta are going to be contributing to function. And what we find is all of these are clustering, all these magenta ones, except for the selectivity filter, of course, are clustering within the interfacial region of the protein, right between the transmembrane and the cytosoluble part. And so this, of course, is where the PIP2 binding pocket is, or PIP2 binding sites between these two helices, where it stabilizes the channel in an open state. And then the G loop, which is the potassium gate that's on the inside, also really strongly comes out. And then there's a really interesting, I think, flexible loop that has specific interactions between the different helices, or between the interfacial helix that binds to PIP2 and then the G loop. So it's kind of an allosteric connector. And then we can even map these colors on here, and we can very beautifully see this hierarchical organization where the top and the bottom drive folding, and then the center drive function, which implies this kind of organization of this channel. So overall, with a mutational scan, we can quite quickly and systematically get to a model of how does the channel fold and how does it function. And we're validating it really against all the beautiful work Carol Deutsch has done on folding, and all the beautiful work many of you, including, say, Rob McKinnon and Colin Nichols, have done on the structure function mechanisms of the inward rectifiers. But then what we wanted to do is try and learn, can we learn something about disease? And not just in CUR 2.1, which of course is this here, but all the other channels in this family, which have many diseases associated with them. And so we did something very, very simple, very, very simple. We just made an alignment across them. I just looked in ClinVar, which is a clinical database for variants, and I looked for where is a frequency, where are there hotspots for disease mutations across all of them, because they all have this really conserved architecture. And I didn't think this was going to work. This was like a purely qualitative exploration, and it blew my mind that all the places that I just talked about directly line up with where there are hotspots for mutations. So that means a couple of things. First, it means we can learn something about disease, and then those diseases, probably those mechanisms are shared. But second, this family is so conserved that how the protein folds is conserved and how the protein functions is conserved. The same parts are involved. And so I just found that mind-blowing, and I think it's really beautiful. But then we wanted to take the next step and do something maybe a little more quantitative to call variants and be able to predict pathogenicity using this data set. So we simply just look at benign mutations, and we can look at benign mutations are rightward shifted on this plot, so more functional than pathogenic mutations, which makes sense. And then we can look across both the surface and the function scores, and we could use that to classify variants and call mechanisms of action for pathogenic mutations. So that means if we had drugs which could be used to rescue surface expression like chaperones versus positive allosteric modulators, we could perhaps treat patients in a very precise way. And then there's, of course, what's called the variant of unknown significance problem, or whereas we don't know what many mutations do because they're not observed frequently enough in the populations, and we haven't seen them enough. And we can start calling pathogenicity and then also mechanism of action for these. And I'm sure that some really more rigorous statistical or clinical geneticists, say Andrew Glaser, who's going to give a talk, I think a couple more, would be able to pull a lot more out of this and have more insights from a disease perspective. But overall, what we're able to do is apply a simple set of multiparametric assays, which allow us to really uncover the mechanistic basis of how this protein folds and how it functions, and then really validate models in the field because we already knew a lot of those answers, but we're able to fill in the gaps, per se. And then we can also use this to push forward clinical genetics. And so in the last section of my talk, I'm going to talk about a project that's really exciting. This is unpublished. We're going to hopefully, we're writing the paper right now, and I hope to get it out in the next month or so, but onto bioRxiv. But I think this is such a cool thing, because what we've done is we've taken the approaches we really developed in CUR 2.1 in a model system. We've started to apply them in a system where we don't know nearly as much, and we certainly don't know how it folds. And I started working on this project about a year ago, or a little over a year ago, working with Kathy Giacomini, who's at UCSF. She's she is Mrs. Drug Transporter Pharmacogenomics. She's been doing this for so long. She started the International Consortium of Transporter Biology, and I had no idea who she was, and I just heard they were wanting to do mutational scans. They wanted to make 300 mutations, and I was like, why don't we make 11,000 instead? And so that's just, it's been this amazing collaboration, and now she's dean here of the school that I'm going to be in, and she's just been such a wonderful champion, and I'm so grateful to Kathy, and she's, yeah. But pretty much, transporters, of course, allow the interchange of molecules across her body, and particularly there are classes of transporters that are polyspecific that move many drugs, and so the same transporter has diverse substrates. And so this drug transporter we've been working on is this cation transporter OCT1 that Kathy cloned a very long time ago. And Polymorphisms with OCT1 are known to change how well different drugs are transported, such as morphine, which will mean that in some cases, patients will get too little drug, so the drug will no longer work in certain patients, or the alternative is true, where they'll get too much drug, so you can get side effects and even patient deaths in the context of some transporters and some drugs. And as with many channel diseases as well, as we sequence our populations more deeply, we find more and more variants for which we do not know what they do. They're these variants of unknown significance. And so we don't know which mutation impacts which transporter. And more deeply, we really don't understand transporter substrate specificity. And let alone for most transporters like these, we don't even understand what they look like from a structure function perspective or how they work or even how they fold. So we set out on this project, and by we, I mean this whole group of people. I've had the great pleasure to work with this incredible senior scientist in Kathy's group, Suk Wah, and two excellent senior scientists, and they're supported by postdoc Chris. And so we took Dimple and made us happy, and to make our OCT1 mutational library, we made stable cell lines. And then with that, we developed a cytotoxicity assay using a cytotoxic substrate that's specific for OCT1, that allows OCT1 to die or cells to die in an OCT1 dependent manner, and that mutations change this. And then we also developed a split fluorescent protein assay for abundance, because we don't have a good antibody, nor do we know where to put a flag tag site. And then from that, we're able to measure for about 11,000 mutations, how that impacts function and abundance. And I won't show you the heat maps, they're massive. But effectively from this, we're able to find this inverse relationship, which intuitively makes sense because cytotoxicity is dependent on transporter function. If the transporter doesn't express up here, the cells live. Whereas if the transporter expresses and functions, the cell dies. And then you can kind of you can imagine drawing a regression here, and that you see the spill out of residues or mutations that are expressed fine, but no longer functional. And so what we did is I calculated the summed absolute value per position across the matrix, the heat map, per position, and that lets us identify which residues are driving stability, so which ones are losing function because they're not expressed, versus which ones that are losing function. And so we can use that to then map on to a structure which parts could drive folding. But we don't have a structure, or we didn't at the time. Now there's a preprint out there from Suk-Young Lee's group on this, but we didn't have it at the time. And so we mapped this on there and onto an AlphaFold structure. So we're able to start understanding structure function mechanisms without any sort of experimental structural biology, because of course AlphaFold is changing the world, and I'm a big believer in that. And so what we can start to do is we can start identify which parts of the transporter are driving stability, and we start focusing on those more closely, because we can see that there's this kind of scaffold of residues that connect the inside to the outside of the cell and that drive folding. And then there's another set of bundles of transmembranes that don't. And so this extracellular domain is made up of a series of aromatics that pack together, and there appears to be a disulfide bond that had been characterized before through mutational studies that we see is very sensitive to. And this looks like to be an independent fold, and it's completely unique to this family of transporters, the SLC22s. And then we find this kind of neck that comes up to connect this extracellular domain to the transmembrane regions. And this is densely packed with a number of aromatics. There's a number of disulfide bonds that also hold this together. So this is probably going to be very rigid in the folding process. There's actually this alpha helix that we can see here that appears to make interactions between the transmembranes and the rest of the protein. And we even looked, and we find that this motif is conserved across all the the transporters in this family. So this is probably a folding determinant that is required. So this isn't not too unlike the poor helix motif in voltage-gated channel superfamilies. So I was very excited about this. Never been described before as a folding determinant, never been described in a way mechanistically to understand how this could even fold. So we're really excited about the potential of doing this across many families, start understanding what is the mechanistic basis of folding. And we also find this interesting network of charged residues, which aren't making an interaction within this AlphaFold structure, but maybe, or model, but maybe they are in some conformational states or in a folding intermediate. And so there's a lot of charge residues. And if we mutate those, they're very, very sensitive. And charge swaps particularly are disruptive. So then we went and did confocal microscopy because we were just measuring expression, not surface expression, to confirm that variants that were loss of function for function specifically make it to the surface fine, whereas those that are these folding variants don't make it to the surface. And this was beautiful work done by a grad student in Kathy's lab, Dina. So then we took this data and we started looking at the function determinants, because maybe we can even start to understand how this transporter functions. So with that, we mapped now the structure and the function, the folding determinants and the function determinants at the same time. We can find that they're really clustering in distinct regions, which is very similar to Kerr 2.1. So that implies there's different regions of these membrane proteins that are guiding function versus folding. And I think this is probably going to be a general phenomena across membrane proteins because there's the hard constraint that it has to be in a membrane and that has to be maintained. What we'll find over time as we do more and more of these across many families and many folds. So we can map this onto the structure. And what we find immediately is we can figure out the substrate binding pocket immediately comes out. This is where in sugar transporters that have similar fold architectures and are the closest subfamily to the SLC22s that OCT1 is a part of, this is where substrates bind. So that's immediately intuitive. That makes sense. But of course, without multiple structural states, it is so hard to learn which residues contribute to a conformational cycle. And I didn't want us to go through the process of trying to solve structures because that'll take forever. And I don't know how to do it. And then we'll have to go down this rabbit hole and other people are going to be way better suited. But instead, I saw this incredible talk by Lucy Delmotte, who many of you probably know about a year ago at a membrane protein conference I was at. And Lucy presented this approach that her star student Darko had developed, which allows them to take coevolution matrices and start with a model, even an AlphaFold model, and start to predict different conformational states in transporters. And because transporters go through a really defined cycle of outward to inward, there are going to be contacts that are made in some states, but not others. And you could use that to then infer those. There's a beautiful preprint out, and I think Darko will have a paper soon that's fully published on this. If you want to look deeper, he's better suited to explain this, of course. But we were able to use this to then predict the entire conformational cycle of OCT1, starting from an AlphaFold model. And you could say, wow, this is just all a bunch of modeling. Is it real? It would be great to have some functional like annotation or validation. Well, of course, we have that functional validation because we have systematically made every single point mutation within this gene. We've identified which parts are involved in function. So if we map the functional determinants onto this, we see very clearly that there's a set of residues that come together to stabilize the inward state, and there's many other things. So we can start to identify a hydrogen bonding network, which are helping to stabilize the outward state. There's many, many different interactions, including this one, which is key to stabilizing that state, as well as many interactions, including what looks to potentially be an ionic lock. Us digging deeper makes us think it's a bit more complicated, but there's a set of interdigitating residues in this interface that help to stabilize the inward state and even a little bit the outward state, it appears. So then we identified 20 mutations because everything before was just a screen that hadn't been characterized before. And we did radioligand uptake experiments with 4 different substrates. This is just one of them that has a similar structure to the cytotoxic substrate we used. And then when we look, they're very closely aligned. We get like nearly a 0.9 correlation coefficient for how similar they are. So this maps really well between our high throughput data and this radioligand uptake experiment. So then we even wanted to go further. We have all these variants. We want to understand really at the core, how do mutations break proteins? So maybe you can do that biophysically. So we went through, and by we, I mean Darko, went through and did 20 different MD simulations and modeling experiments, coming up with whole conformational ensembles for 20 mutations. And then we were able to just ask the question of how does the distribution between the outward state and the inward states change? So when you add a substrate, this gets pushed into the inward state, and you'd imagine the substrate comes off and then it'll recycle and then restabilize the outward state. And we're able to find mutations which create energetic barriers. So this is one of the loss of function variants that creates a large energetic barrier. So the binding of substrate can no longer cause that conformational change. So you're not going to get the uptake of that. So we went through for all of these mutations, and using all the data available for us, we identified the biophysical and the thermodynamic determinant by which a mutation is breaking the transporter or even making it work better. So we're able to be really mechanistic about this sort of work. So finally, we wanted to look more deeply and say, what can we learn about human variation? So we just asked the question first, what percentage had been characterized before? As it turns out, there are about 300 variants that have been observed across 150,000 or 200,000 exomes that are present, that are available online. Of those, 20 had been characterized before, so those are the ones in orange, and all the other ones hadn't. So, we now can say something about the vast majority of mutations and whether they're loss of function, and if they are loss of function, what the mechanism is of that. But then finally, we wanted to ask the question, does this DMS correlate with the impact in real patients in human physiology? So we took this data, and by we, I mean Sukwa, when she went down to Stanford as a visitor in John Whitty's lab, who does a lot of patient health records comparison with genetic databases. And so we looked at the UK Biobank, which, if you don't know, is this database of hundreds of thousands of genomes associated with patient health records. So you can ask the question, how does this variant associate with different, say, blood tests? And OCT1 function is known to change LDL levels. And so there's an increase in LDL levels when OCT1 is broken. And what we can find is that we find poor functioning SNPs. This is wrong. I need to change this one word. But poor functioning SNPs are strongly correlated with increased LDL levels, whereas normal functioning SNPs are not. And what that means is we can really use this data to say something about how mutations change physiology of patients. And so going forward, one of the projects we're working on is doing mutational scans now across all the FDA prioritized drug transporters. We're in the process of doing many of these, and we'll use these to really discover basic biology. I'm really excited to explore how substrate specificity evolves across transporters and how do pockets change to allow different substrates in. And then also we'll work with collaborators who are going to take this data and compare it with patient health records to really guide precision medicine. So potentially even be able to say, okay, this patient comes in, they have this variant, what drug should they take? How much of that drug should they take to minimize side effects? So with that, I've shown you how we can use these multi-parametric assays to learn something about CUR 2.1, how we've been learning about OCT1, and how we can even expand to different perturbation classes. And now in my lab, what we're really doing is we're developing other sort of perturbations so we can better explore the sort of variation that we naturally make in us. And then we're also exploring how those change proteins, membrane proteins, mechanistically. And so for that, we really need a toolkit. And so we've been working on that. So we can have general readouts of, say, folding and trafficking, a GPCR signaling assay, which I would love to talk more about, but I think I already have too much data. So we have a whole set of screens we've been doing on GPCRs where we're trying to do real pharmacology and do full dose response curves and these sort of assays. And then also trying to measure how does trafficking change in the context of this, because lots of GPCRs and other membrane proteins internalize, and many, many other assays. And this is a huge team effort. And we're going to use this toolkit to really study how perturbations change membrane proteins. So how do mutations and stimuli and CRISPR and genes all work together to change mechanisms of membrane proteins, which we're finally getting back to, hopefully, learning how membrane proteins underlie our physiology. How do they break in disease? And how can we better treat them with pharmacologies? And finally, I want to thank you for listening to me ramble, acknowledge the many, many people who've been contributing to this work, some of who I did not get to fully highlight their beautiful work. The Fraser Lab at UCSF that gave me a home in the pandemic, and then also has really given me a huge support to launch myself. And then many collaborators at UCSF and across the world, some of whom I can't wait to share more of the work we're doing together, and my PhD mentor, who gave me tremendous amount of freedom and support to really do whatever I wanted, and of course, my funding. And finally, if you're looking for a job, we're hiring at all levels. And if you want to join us on this incredible journey of science, I'd love to talk to you. So yeah, thanks.
Brilliant. Thank you very much, Willow. Fantastic. So we've got a number of questions in the Q&A we can jump into. If anyone has a question and they'd like to vocalize it, they can raise their hand. Whilst we wait for anyone there, maybe I'll start with the ones in the Q&A. So Damien Bell asks, in the indel libraries, the count tails off in the C-terminal end of the protein. Is there an explanation for this?
It's sequencing. So the way we make sequencing libraries is this is really getting into the weeds. So we use these transposons, and the transposons don't like the end of DNA. So we try and include even more of the DNA, but like at the C-terminal side, but for some reason they have a bias away from like inserting sequencing adapters on the C-terminus. So it's not the library generation. If we do Sanger sequencing, we don't see it, but it's more, it's like how we sequence things. Yeah.
Got it. And so Jian Gao, who's a student in Colin's lab, in Colin Nichols' lab, says, nice talk. Are there some special tricks for guaranteeing a good coverage of the diversity of the library in the cells?
Yeah, so I think for that, all you have to, like, you just have to, there's a lot of tricks that we've developed, and we're happy to share them. If you want to, we can send our protocols your way. Like, because we're always happy to help other people. But effectively, what it is you have to make sure you're always maintaining diversity. So we know what the efficiency is of getting DNA into the cells. It's something like 5% of cells get DNA in the end, and the rest we select away when we're making these cell lines. And so you just have to scale it massively, effectively. And it's not that hard. I mean, it's not that massive. So for like the Curt 2.1 work, we did like 2 10-centimeter dishes worth of transfections, and then that gives us enough coverage. But you just have to make sure to calculate that you're always having enough coverage. And I always shoot for about 100x or even 1000x the number of cells as the size of your library after you account for kind of how much is dying and things like that. I don't know if that's helpful, but it's pretty much just make sure you have a lot.
Do you see a correlation with the size of the inserts compared to how many cells you successfully transform?
I haven't tested that really rigorously. I would say when you get to a bigger size, then that means your library is bigger. So so it's not so much your coverage goes down because of that. And like one of the tricks that we're trying to work with a really, really big gene that's like well over 3,000 amino acids just to like really push, or 4,000. And it's like to make our lives really difficult. And like in that case, we're going to break it up into like 8 parts and then make sub-libraries. These serine recombinases, which are what is used to get DNA into the genome, those have been in the literature. People have described getting things like 40 kb in without a problem. I can let you know how a 17 kb piece of DNA gets in like a matter of a couple months, and let you know how much of a struggle we have. So, yeah.
Damien has another question. So he noticed that when you showed the sequence alignment for the transporters in the neck region, that FLIC-T1 seems to have 2 or 3 significant differences. Non-conserved amino acids, a cysteine at the end, and a glutamate halfway through. Is this important? Does this suggest deviation in the conserved region for FLIP1? And he says some nice things at the end because he's a nice bloke. Fantastic work and techniques, lovely stuff.
Thanks, Damien. Yeah, I haven't dug deeply enough into this, is a short answer. I think it probably does. I think it infers that there are probably some differences. What I would really love to do, and I just haven't gone through this because I just haven't done it yet, is make a much more systematic alignment. So take blast, of course, the sequence that we're starting with, and just take everything and look at covariation and see in the context of FLIP-T, because I've noticed this as well, ask the question, are there covarying residues that imply, that there are new contexts that are being made and other ones that are being lost, and try and also look at that, of course, in the AlphaFold model and see if that tells us something, because I think that's a definite important question. I still think this alpha helix and then a bunch of residues coming off and making interactions with the TMs, I think will probably be true. But what those specific contexts can be made, of course, may diversify, because a lot of these transporters aren't under selection too, which is really fun because they then have like room to move sequence wise. So yeah, that's a great question. And I look forward to having a better answer. that's more, satisfying.
And Rohit Kumar says, thank you for the very nice talk. From the heat map data, you have mentioned that the ER motifs, I think this is for 2.1, are less sensitive. Sorry, I missed that. Would you please be able to explain that once more? Also, how this observation unfolds in conventional trafficking of the channel. And then a second question was, what was the training data set used for the machine learning based MD simulations?
So I'll answer the first one. So first, I'll answer the second one second, obviously. But in the ER motif, I found this interesting because I went in thinking that we'd see the Golgi motif and the ER motif, and the ER motif would completely break trafficking. And the ER motif is a diacitic motif. It's ENE, and then there's like some aromatics before it, and this has been seen in other ER export motifs. And so the other trafficking motifs are in the hydrophobic cores and in or around the cores of the protein. And so if you mutate those, I think we disrupt folding as well. So you see a much more significant phenotype. Whereas in this diacitic motif, I don't think people have solved a structure of the trafficking partners and what actually binds and gets it to the cell surface. But my guess is that having two allows a higher affinity for whatever comes and binds. And if you remove one acidic residue, then it can only bind one. And so you'll have a lower affinity, but some will probably still bind and some will still make it to the surface, if that makes sense. And then what we found that I thought was very interesting there, in case you missed it, is that if you make a third acidic motif in between these two E's, it actually boosts surface expression even higher, which kind of implies that probably it's like I think it's like a sequence search sort of thing, that you just have more there and maybe it binds better. But these are all unstructured regions of the protein. And I would make a strong argument that in the membrane protein field, including channels, we kind of like chop them off and ignore them. And I think there's a lot of really interesting stuff going on in these regions for understanding trafficking and understanding the logic of it. So effectively, the idea is that there's chaperone that I'm forgetting the name of right now, I apologize, that comes and binds in the ER and recognizes this motif and gets it to the cell surface. And people have seen before that if you remove that, it can no longer get to the cell surface. I hope that.
So Willow, are there tri-acidic motifs found in nature, or in 1 week have you beaten billions of years of evolution here? Right?
That's a really good question that I don't know. I think like there's this kind of funny thing with evolution, right? Is that you're not necessarily trying to get the thing that expresses the best. You're trying to get the thing that works well enough. Right. But I would imagine there's a utility of having higher expression in some context, but like having too much CUR 2.1 at the cell surface could also cause disease phenotypes, of course. So like, there's this tuning where you don't always want the mostest, but I'm not sure. That's a really, really good question that I should go into and try and answer. Especially when we do this in other backgrounds where we have other diacitic motifs.
Rohit asked about the training.
Yeah. So the way that was done, and I'm going to do a really bad job of explaining this because this is like Lucy and Darko's, and there's a beautiful paper on this. But effectively, what they do is they generate an alignment, and then using that alignment, they find co-evolving residues, and they then use those co-evolving residues to bias the MD simulations to make certain contacts. And then they use the co-evolving residues to try and push the protein together, and then they use the MD simulations to relax that structure. In terms of what the training data set, I'm pretty sure it was done based on the AlphaFold model alone and starting there. But they have this paper where they did it in the context of sugar transporters, where they much more rigorously kind of validate it in a system where they have many, many structures present. And I encourage you to check out that paper because I will not do it in any service at all. Okay, awesome. Colin Nichols points out, too much CUR 2.1 causes short QT syndrome, just kind of to that point. Sorry.
Maybe I could ask a question. So for your transporter stuff, which was really interesting, if you do the cytotoxicity assay with a different cytotoxic chemical, presumably you would pull out different mutants that might help you to map the substrate recognition site, right?
Yes. Yes,
Cool.
that is like exactly what we want to do next in these studies, and do it like try and do it from different genetic starting points, and especially because the vast majority of these mutations are affecting folding. So like those are probably not going to be interesting in the context of this assay. And if you look at a substrate binding pocket, it's not one residue, obviously, that's causing this. It's many, and it's the interaction between those. And in evolution, it's the changing of all these this pocket that allows the binding of substrates. And what I really want to do, and this is like, to me, one of the most exciting projects that I just want to like chase down and try and figure out how to do is how do we make higher order libraries where we make multiple mutations in a programmable fashion at the substrate binding pocket, and then cross that with different chemicals, and then try and start, say, in this family specifically, where there's cation transporters, and diversified, there's also Zwitter ion transporters, and then there's anion transporters in the same family. And you could imagine trying to ask the evolutionary question, like, how does a Zwitter ion transporter become one or the other, you know? And there's clusters of both in sequence and us that have redundancy and then some specificity within these different classes of chemicals. So that's like, to me, such an exciting question. Yeah, so that's the idea.
Well, we're at 58 minutes past, so I guess we wrap up now, Willow. Thank you so much for kicking us off. That was a really exciting start to the series. Look forward to continuing. And we'll meet after this, right?
Yeah, sounds good.
Okay.
Thanks so much, everyone. Have a good rest of your day.
Thank you.
Thank you, everyone, for attending.